Uh, so today, this Saturday, we are going to be hearing about genomics and various aspects of using genomics data in the context of medical conditions, research, and preventive health. That is discovering and manage disease risk. And the expert today is Professor Ramesh Hariharan, who is the CEO of Strand Life Sciences and an adjunct professor at Indian Institute of Science, Bengaluru. He co-founded Strand Genomics along with three other colleagues, collectively called as the Gang of Four, that focuses on mining and modeling challenges in life science and drug discovery research. Ramesh did his BTech from IIT Delhi, PhD from Courant Institute, NYU, postdoc at Max Planck before joining IISc. He's led a successful entrepreneur journey, and he has been deeply involved in the creation of GeneSpring, an integrated data analysis and interpretation platform for all types of microarray applications. And also Grammatica, a natural language processing engine that drives biological interaction database in GeneSpring. He is a fellow of the Indian Academy of Sciences and the National Academy of Engineers, recipient of the Devang Mehta Award for Innovation in IT from the Government of India. So among several other things he has done, I want to just pick up the fantastic aspect that he's written a popular book. It's called Genomic Quirks, The Search for Spelling Errors. I've read this book. It's there in our library here. This is a book of real stories about the search for genomic spelling errors that have stark consequences. So I really look forward to hearing from him. Moderating the webinar is another expert, Dr. Shweta Ramdas, from Center for Brain Research at the IISc campus. She earned her PhD in bioinformatics and MA in applied statistics from the University of Michigan. After that, she did a postdoc work at UPenn, and she joined Azim Premji University before coming to CBR. So, I mean, we have the perfect combination for today's webinar. And I request the audience to engage with questions through the Q&A box, not through the chat, okay? So look forward to this talk. Over to you, Ramesh.
Thank you so much, Professor Naran, and to all the organizers. Thank you to the audience for attending. I've been, I and of course we at Strand collectively have been working on genomics for a long time, and genomics is a story that's really evolved dramatically over the last 20 years. I want to convey some of that evolution to you, as also where we stand and how this provides an additional tool that can do very interesting things that we could not do earlier. So let me start with the genome. It's we all know what it is at some level. Abstractly speaking, it's ocean of characters inside us, 3.2 billion characters, and they're all A's, C's, G's, and T's. And not just that, we in fact have two copies of these 3 billion characters. One set of copies comes from one copy comes from our mother, one from our father. So we essentially have 6.4 billion characters in all, right? And these are organized into 23 into 2 chromosomes. So what that means is that the one set of chromosomes, 23 of them come from a mother, one set of chromosomes, 23, another 23 comes from a father. And I like to often depict it like this bookshelf. There are these books on a bookshelf, 23 pairs of books. Each book, if you open it, it would be just a unintelligible ocean of characters. And there are in each pair of books, like you can see that this book is sort of larger than this book. There are more characters in the chromosome 1 book than in the chromosome 8 book. because these chromosomes have varying sizes. But other than that, the content is, this stuff. And let's dig a little bit into this content and get a sense for what we can do with it. So if I go into one of these books, I'll just see this. And there's not a whole lot of sense you can make of this, right? And one important thing to note is each one of us is different in this content. Somewhere or the other, there are differences between you and me. And those differences tend to be about 1,000 characters. Which 1 in 1,000? 1 in 1,000 is a rough, distribution across the 3.2 billion characters, but which 1 in 1,000 can differ? You and I may be different in a certain 1 in 1,000. Somebody else and I may be different in a different 1 in 1,000, etc. So sprinkled amongst in this ocean are these differences, or what we call variants. And these variants, of course, are important in some form. I mean, after all, they make me different from you. I might have different appearance, a different skill set, etc., partly on account of these variants. So that leads to our fundamental question, right? You and I are different, and nature is constantly tweaking and creating new individuals with more differences. So what if nature tweaks? Let's say I pick out a character here, deep inside here. What if nature were to tweak this character and change it from a T to a C? And as I said, there are 2 copies of the genome that we inherit, right? One from each of our parents. And let's say nature tweaks this in only one of the 2 copies. Now, what happens? Does it make a difference? It turns out that you can tweak almost any character here, and nothing might really materially change. But there are some characters, if you tweak, they will have some serious health impact. So let's focus on this particular character. And I've also mentioned the name of the gene it is in, and it's a gene name that you might recognize. So genes are, some part of the real estate of the genome is occupied by these so-called genes. They have certain properties in the sense that they carry recipes for the creation of new molecules called proteins. So this is the real estate occupied by the BRCA gene, and somewhere in here nature has tweaked this character. Then what happens? It turns out that if we tweak that character, and it indeed was naturally tweaked in this family, and in this family there was pretty much everybody had some form of cancer or the other, or many people had. And some people had breast cancer, some had other forms of cancer, etc. Occurred in their 50s, 60s. And it turns out that this character change, it has this complex nomenclature, which you need not, don't worry too much about this. The fact is that T on the previous slide changed, and that little change increases a person's risk of breast cancer by about 6 times and of ovarian cancer by about 30 times if you're a woman, assuming you're a woman. So, and for other cancers by lesser amounts. Okay. So the point being, a little tweak at that little character T amongst this ocean of 3 billion can increase your risk of cancer substantially. Let's look at another example. In this example, this is a different gene. And somewhere in here, there's a character G sitting, again, very nondescript. You can almost hide and you would never know that it is important. But if nature tweaks that character, the consequence is even more dramatic. While this person is in the mother's womb, the structural developmental program that drives the creation of a human, a fetus, and into a full baby and thereafter, that goes wrong quite seriously, in the sense that there's major structural and developmental changes that happen. For example, talipes is a word for club feet. The skeleton is deformed. The brain is not fully properly formed. The amniotic sac and the fluid that is around it has abnormalities to it, and all of that leads to a high chance that the baby will be born with a lot of complexities. And in this particular case, and therefore it makes sense when you get to that point to terminate the pregnancy instead of continuing with it. And all because this single character was tweaked. Now, one important thing to note though is such serious consequences usually happen when this tweak happens in both chromosomes. Remember, we have two copies, one from each parent of every genomic character. And these very serious consequences often happen when both of those characters are tweaked. If only one is tweaked, then the other still protects us. But when both are tweaked, then you can get into this sort of a situation. That's not always true. There are single character tweaks also that are problematic. But a general rule is that these sorts of extreme situations often require both characters to be tweaked. While the previous example that I gave, this one, it's only in one of the two copies of one of the two chromosomes. And you're still born well, you still continue to live a proper life, but your risk of disease goes up substantially. So if you could look at it, and that happens with the change in only one of the two copies of the character. So if you look at this as two extremes of genomic character tweaks that can create issues, one allows you to live a long and healthy life, but your risk of cancer goes up, and therefore your life expectancy might come down substantially. On the other hand, it might be absolutely nipped in the bud by something far more serious. And these tweaks of character sitting inside can cause all kinds of shades in between. Things that show up when you might be born perfectly fine as a baby, but might show up in your teens, in your 20s, in your 30s. It might be degenerative. So it might set in your 20s and rapidly degenerate through your 30s, 40s. Entire gamut, entire spectrum exists. Right? And that leads to these questions. Now that I've set up the background, as I said, each of us differs from everybody else in like 1 in 1,000 characters, right? And those are called variants. So how can I know my variants? I want to know my variants. Where am I different from most other people? How do I get to know those places? Once I know those places, well, even before I know those places, how good and robust is this process today? Is it erroneous? I mean, can I trust what I'm told, or should I take it with a pinch of salt? Suppose it is trustworthy, suppose it is robust. How can I use it now? Can I predict my risk or that of my family for various diseases? Sorry, I missed the S at the end, but for various diseases. And if so, then what action can I take? Right? Those are sort of the questions that arise in this context. So let's explore some of these questions. I mean, this is an ocean again, and it's hard to do justice to everything, but we will at least touch upon a few things in the next 45 minutes or so. So let's start with, how do we read the genome and get hold of our variants? What does that process look like? So the story began in the year 2000, or just before that. Until before that, there was little we could do to really, if I wanted to understand my genome, there's little that one could have done. Around the late part of the millennium and around 2000, the first human genome was published. And that process was not a simple process. It required massive national scale as well as private projects running into billions of dollars of investment. Thousands or maybe even more scientists working on it, all on just taking like one person's genome or a handful of people's genomes and trying to get, sequence it. But once that happened, and that happened at tremendous cost, as I said, both in terms of time, effort, money, etc. But once that happened, it opened up some interesting possibilities. And for the first 15 years of the millennium, what we could do in terms of reading the genome was to probe it at certain locations. Because we knew this entire sequence for 1 or 2 or 3 or 5 people, that which came out through the human genome, the very onerous human genome effort, which is not repeatable. So you couldn't have repeated that with every person on Earth, right? If you wanted as a consumer to say, I want to get my genome sequenced, that was not possible. But what became possible as a result of the Human Genome Project was that we got to know this entire sequence, what you see, um, the sequence of ACGTs. Now, whose sequence is that? Is that mine? Is that yours? No, it's some 4 or 5 people and their consensus sequence, so to say. So it represents the sequence of the human species at some level. It's a representative sequence, not an individual sequence. And that representative sequence we call the reference sequence. And now, from the year 2000 onwards, if I wanted to do something about my genome, I could not really get this level of, this entire sequence for myself. That was not possible. What was possible was that I could probe my genome at a few predetermined locations, like the ones marked in red here. Through this method, this technology was called the microarray technology, and through that I could probe the genome at a few select locations. And the locations initially started with a small set, but over a decade or so, it grew to almost like you could probe it at 1 million locations in your genome out of the 3 billion that's there, right? Maybe a couple of million even. And when you probed it at these locations, you would get your characters. So I could have, while the reference sequence, which is some 5 people's consensus, has a C, I might have a G here. And so I could figure that out. So I could figure my variants out at these select locations, which had to be predetermined, right? It's not as you could come in and say, hey, I want to get my genome done at these million locations, and somebody else says, I want to get mine done at these other million locations. No, not that. So a set of locations was predetermined, and at those locations, you could actually figure out what your characters were. And that was doable in, let's say, a few $1,000s or something like that, over a period of time, coming down to a few $100s. And then the question became really, how useful is this to probe at a few locations and get those values down? And the bigger question it leads to is if I were to offer this as a service, then which locations should I pick and offer you? Okay. And that leads to this notion of what are called common variants. Now, the locations I should pick, if everybody is likely to have the same character at those locations, then there's no point picking that location. I might as well tell you the value you're likely to have at that location. So we need to pick places where many people are different. Some people have G and some people have maybe C there, and enough people have G's and C's. If only one person on Earth has C and everybody else has a G, then when you pick that location and do it on most people, they're just going to get the default answer. It's not very efficient cost of use of funds to do that. So typically what you do is to pick these locations where there are enough people with a G and then there are enough people with another character, let's say a C. And this is an example of such a variant. And what you see is for different population groups, we now know for many characters in the genome, how many people might have one character and how many might have the other value at that character. So in this particular case, South Asians, the reference character, whichever was the reference character, let's say here, that's a C, is there in most people, but about 45,000 out of 91,000 have the alternate character, which is a G, let's say. So about 49% have a G. Overall, if I look across populations, about 33% of people at this location have a G, while the remaining have a C. Right? So typically one would pick these sorts of characters so that one can get enough variant information out when you test yourself, right? And so all of this testing for the first couple of decades where individuals would do their genomes was done with this method, essentially measuring common variants, where this number, 33%, is a substantial number. It's not like 0.001%. It's like at least 5%, let's say. So such variants are called common variants. And such common variants are what we know about, and what you can guess about such common variants is they're unlikely to have a major solo impact, right? So many people have these variants. And if this variant had an impact on health, that G was harmful and C was okay, then 33% of individuals should see some sort of disease phenomenon. But diseases from purely on account of genetics is particularly on account of single variant is rarely that common. So typically such common variants have very limited solo impact. Usually they are likely to act in combinations. And in combinations, they have very powerful impact. So many such common variants act together and they have very powerful impact. But a single such solo variant acting in solo is unlikely. So net effect of all this was that for the first 20 years of the evolution of genomics, or the first 15 years at least, what we could measure were these common variants. And with these common variants, we could only assess impact where large combinations of these common variants could do something together, but we couldn't assess individual impacts at all. To give you just a sense for how predictive this can be, even if you take the height of an individual, right? The height is something we consider very predictive, right? I mean, if you were tall, you would say that your children are likely to be tall. And extensive research has happened on height. And today, we can barely predict the height of an individual. So studies have been run on 5 million individuals using these common variant microarray techniques. We have identified through all of that analysis about 12,000 characters in the genome, which are predictive of height. And we believe that these are the only 12,000 that govern the height in the genome. But together, when we predict the height of a person, these can account for only 40% of the variation that we see around us. The remaining 60% of variation is not accountable by these 12,000 characters in the genome. In other words, we can only predict, I mean, if I were to use common parlance, maybe to the tune of 40% we are able to predict. That is for Europeans, even lesser so for other ancestries. You cannot predict the height of an individual using a handful of characters in the genome at all. And even predicting like using 12,000 variants, we struggle with being able to do that to a large extent. So if I were to apply this on an individual level, what this would mean at an individual level is that the predictions will have fair amounts of error. If I apply it at a large population level, I will still get statistically decent predictions, but at an individual level, these won't be reliable. So such common variants and using combinations of those to make predictions is something that's not easy for us today to do. And to therefore, for us to individually apply this in our lives and say, okay, I've got these 12,000 variants, and therefore I can predict something about my risk of getting diabetes or having some other disease, that gets very hard. Okay? So that is what we could do for the first 15 years. And things have changed dramatically over the last 5-7 years. Because we realized that there are a lot of rare variants in our genomes. What is a rare variant? So here is an example of a rare variant. It's in the same gene that I pointed out earlier in my first example, where which leads to a very high risk of cancer. This particular variant is found, as you can see, in very few people, right? In South Asians, out of 86,000 people tested, only 3 have that variant, which is a pretty small. This is just the ratio of this to this. That's a very small number. Overall, across the world, 0.0013% of individuals have this variant. But those who have it, have a much higher cancer risk. And that cancer risk is driven almost that elevation of a large chunk of that elevation of risk is single-handedly driven by this variant. So these things we call monogenic. Monogenic means that a single character in the genome, sometimes it's two characters, a single or two characters in the genome can drive risk substantially. And such characters are things that we could not assess using the microarray common variant technology. But over the last 5 or 7 years, with a sea change in technology, we are able to actually now assess these very rare variants. Rare variants are things which at a population scale are very rare, but you'll be surprised at how at an individual level, every one of us almost has, I'll come to the statistics, but a large fraction of us have a rare variant of relevance. How do these rare and common variants together? I'll come to examples and what this means for each of us. But now that I've introduced this notion of rare and common variants, if I take any disease, heart disease, diabetes, etc., what fraction of the risk for these comes as a result of common variants and the large number of those collaborating to increase risk versus these monogenic rare variants that across the entire population are rare, but every individual will have their own rare variant that might increase the risk. Not every, but a number of individuals, a good fraction of individuals will have their own private variants that might increase risk in their own form. How is the risk divided between all of this? So I'll give you one example. This is autism. I could have picked diabetes or something like that, and the picture would look a little different. Diabetes is very complex because it's not only genetics, it has a big lifestyle component. A large chunk of diabetes can be managed through lifestyle, as we know. Autism is a little different. There's not so much, nothing you can do. It sets in early, right? And there's nothing from a lifestyle perspective, very unclear what you can do. And therefore, I picked this as an example of what fraction of the risk do we know today comes from common variants and what comes from rare variants. And what we know today is that only a small fraction of the risk, right? About, you could say 6 to 10% comes from variants that are very rare versus a large chunk of the risk comes from common variants. And therefore, it would make sense to focus a lot on common variants, except for the fact that they operate in large groups, and to decipher what they're doing in large groups requires such large population sizes that we don't have that knowledge today. Maybe we'll never have that knowledge because it enters the realm of complex phenomena which are very hard to predict. On the other hand, the rare world, we are relatively much better off in both in predicting what that variant might do. We're not 100% there. We're perhaps not even 50% there, but we are certainly few tens of percents there, which is a healthy percent for us to be able to now today apply that in practice. And the sea change that's happened over the last 5-7 years is that our ability to sequence and measure these rare variants has become phenomenal. And that leads to the notion of sequencing. How do I sequence my DNA so that I can identify not only the common variants, but also the rare ones? And rare, as I said, is relative to the population. 1 in 10,000 people might have it, but the person who has it might have a serious consequence. And looking at it from the individual's perspective, many people will have their own rare variant. So it's rare doesn't mean uncommon in some sense of the word. It just means that every variant by itself is rare, but many individuals will have their own rare variant. So in that sense, it's common. So with that, let me come to the sea change in technology that's happened over the last 7-8 years that we've all been working on, which is sequencing. So let's say 2015 onwards, even though it's a little prior to that, but really in common practice, 2015 onwards. It's very different from the microarray technology that I talked about, which is that you're not just probing the genome at a few select locations. You're really getting every location. And you don't have to make up your mind in advance, I'm going to probe it at these locations. You just sequence broadly. And the way that sequencing happens is that you take, let's say, whatever, blood or saliva or any tissue from your body, and extract DNA out of that. And the only little unfortunate thing about this technology is that you can't just read that entire DNA, that DNA, if this is the sequence of characters, you can't just read the whole thing. You have to chop it up into pieces. So once you chop it up into pieces, the little pieces, each individual piece, you sort of get the entire sequence, ACGT sequence within that piece. And these pieces are typically about 150 in length, while the whole genome, as I said, whole thing is 3 billion, but it's broken into chromosomes. So the largest chromosome might be 1/10 of that, so 300 million, but we can only read chunks of about 150. And so now that leads to this whole complexity of, I've torn this little book of characters into tiny shreds of paper, and how do I put it back together so that I can understand what my variants are? And we can do that, and I'll come to that in a bit. But I want to tell you why this is dramatic, because the cost of this sequencing was, as I said, in the turn of the millennium, $100 million or more. And it's through advances in biochemistry, molecular biology, optics, computing, algorithms. I mean, everything had to come together to make this come down. And today we are at the cusp of it being just $300 to sequence an individual's genome. Right? So this is a dramatic change. And with the sequencing, we can identify all common variants, we can identify rare variants. And with the rare variants opens up, because they are more, their impact is more predictable, it opens up an whole avenue of, now can I predict what these things do for my health? So this is a good point to break, and I would like to invite Shweta Ramdas, who's moderating this session. Shweta is a researcher professor at the Center for Brain Sciences at IISc, and has made a name in lipid genomics, understanding the genomics of heart disease and lipid metabolism, lipid genetics, so to say. And I'll invite her to sort of step in at this point, and we'll have a little conversation before we resume the talk again. So over to you, Shweta.
Thanks, Ramesh. Can you hear me okay?
Yes, perfect.
Yeah, okay. Okay. So, yeah, I thought I would start out with your discussion about microarrays versus sequencing. I think most people hear about genomics through companies like 23andMe or Map My Genome, or different sort of industrial partners that tell us that we can get ourselves genotyped, and then that will inevitably sort of tell us about our wellness, diet, etc. So could you tell us which of these two technologies these companies use? And the second one, sort of a similar question came up in the chat, which is, what are the costs of sequencing versus microarrays at the moment? And how well do these measurements really help us with things like wellness?
Yes. For good reason, as I mentioned, for the first couple of decades of this millennium, the only thing we could do was microarrays, and therefore a lot of research happened using microarrays, but also a lot of commercial activity around trying to deploy microarrays in a more layman situation, trying to see whether one can predict risks and do something useful. And I would reasonably conclude from that these 2 decades of experience that that effort on the research side has yielded a fair amount of insight into discoveries, fundamental discoveries. But on the deployment into healthcare side, it's been largely a failure. And the reason for that has been exactly the reason I mentioned earlier. What you can determine using microarrays are common variants. And these common variants operate in such large, they don't operate solo. And so they lead to these complex phenomena where we are not very good at very multifactorial. If something is caused because of 100 or 1,000 or even 12,000 things operating together, to make predictions using those, we are just not, we're not equipped today. And that day will come as data sizes grow, or may never come. We don't know. That jury is out, but certainly not today. For example, I'll just give you this example, right? This is a 23andMe. So many people know about 23andMe. They broke ground in the US with this microarray test that would do many things. They use microarrays and use common variants, which are reasonable for ancestry, which was one of their popular applications to help you figure out what your ancestry, which perhaps your ancestors came from. If you're in the US, might have come from Europe, certain parts of Europe, or from Eastern Europe, or even from Africa and things like that. That was good, but not so good for health predictions. For instance, this would say that I'm lactose intolerant, right? Likely intolerant. And if you look at how it would say that, it would look at this one character where I have a G in both my copies. But when you look at that character, pretty much everybody has a, South Asian, 97.9% have a G. I mean, you might as well close your eyes and say that you're lactose intolerant if that's what the statistical background of the population is. So these things did not yield, anything particularly interesting that was useful. So, but with sequencing, that is changing. So that's the, I would say that would be my take on microarrays with sequencing. Sequencing is the future, is the current and the future. Microarrays are largely on their way out, except for very, very special applications. And in terms of costs, which was the last part of your question, sequencing was impossible, right? In 2000 to 2015, very expensive. Microarrays came down to like a few $100s, maybe even down to $100, $200. Today, they're around that, while sequencing has steadily made its way down, and it's down to about $300 a person just to sequence. But often you don't sequence the entire genome. You can do a lot by not sequencing the entire genome, but by sequencing just parts of it, relevant parts of it. For example, just the so-called genes. And that can come down even to like 10,000 rupees, 15,000 rupees, right? I mean, that realm of things. So that really makes things within reach of a reasonable section of the Indian population to actually see what health impact it might have.
Another question that I think there's a lot of interest in the application of genomics to health,
Right.
Understandably. And one question that came up is, how do you know, or other ways for us to understand which diseases can be modified through lifestyle changes through the environment, and which diseases are more likely to be sort of more hard-coded, so to speak?
Yeah. So I will go through some examples in the rest of the talk, but a thumb rule is diseases that occur early on are more likely to be, of course, completely have a much greater genetic component than a lifestyle component. Diseases that are age-related will have, which occur over a period of time, will have, I shouldn't say will have, often have a strong lifestyle component, which is good news, which means that you can do something with it. Something that's purely genetic, harder to do something with. Something that is has a lifestyle component, you can alter your lifestyle. Most common diseases that we know of tend to be heavily combinations, have a very large lifestyle component. And that makes the application of genetics to those somewhat limited. But there is enough that's there today that is very genetically driven, very monogenically driven, even more genetically driven, very monogenically driven, and where we can do something about it to change things. So that's going to be the latter part of the talk, which I'll come to. But it's a large spectrum, it's evolving, and I don't have a simple yes/no answer to that, other than there's a full spectrum out there.
There's actually a lot more questions, but this might be a good time to move on to the next part and sort of come back to a lot of these, which we can also address at the end. So would you like to go on?
Okay, so I'll continue, and I'll hopefully answer some of these questions in this process, and we'll take more questions towards the end. Okay, so the once you've sequenced your DNA, right? What do you do? What do we do if we were to sequence it? You asked us to sequence your DNA. What would we do after we sequence? So remember, when you sequence, you have to tear up your book into little pieces, right? That the little shreds of length 150 or something like that. Let me just show you that picture again quickly. So you've got to tear it up like this, and so you get these little pieces. So these little pieces depicted here in these, each orange bar refers to one of those little scraps of paper, right? So what you do with that is you compare these little orange bars to the reference genome that you have, which is somebody else's genome, done 20 years ago maybe, and you compare and see where the differences are, and you identify these differences. Here, all the pieces of the orange stuff all comes from, let's say I'm getting my genome sequence, then the orange stuff is all the shreds of paper that come from my sequencing, while the blue is the reference sequence for the entire species, which is really some 4 or 5 people's sequence that was done by the Human Genome Project 20 years ago, let's say. Then I just compare all my shreds of paper with the reference sequence, and I see that, at this character A, all my shreds of paper saying I have a T, while the reference sequence is saying there is an A. So that tells me that I'm different. So this is a variant here, right? So now, these are the variants, and sometimes these variants, you have to be a little careful. All my shreds of paper may not say the same thing. Some might say there's an A, some might say there's a T, because again, I have 2 copies of the genome, right? One comes from my father, one from my mother, and my mother's copy might say there's a T, my father's might say there's an A, so I've got to be cognizant of that. So I have a variant now in only one of my 2 copies versus this previous variant that's there in both copies. So with that, so I make a list of my variants, and then the question arises, what does this variant do? And that's a big, a complex process, which I may not tell you too much about, but let me give you a couple of examples of diseases and what the picture looks like for them. So here is a case where this was a pair of siblings who, in their 20s, their heart became really weak. I mean, could barely pump. That state is called a state of heart failure. And when we took their sequencing data, their scraps of paper, these orange things, and laid them out against the reference genome, what we saw is that in one of their 2 copies, so one copy comes from their mother, one from the father, so one of their 2 copies, these 2 characters AG were just missing, right? And when something like that happens, when characters are missing, that usually, this would be in some genes called the DSP gene, which has an important structural role to play in the heart. And when characters go missing, then that cripples the whole gene in some serious form. And that we could convincingly establish that these 2 characters being missing is why, these 2 siblings, their structure, the heart structure was completely compromised. And one copy was sufficient, right? Even though the other copy was good, it couldn't protect. So several genes are like this, that one copy has something missing or something that's changed, then it can't protect, while in several genes, you need both copies to go bad. All kinds of, both scenarios exist. Another case where you saw this 2 character deletion here. Now this is actually a much, much bigger, it's a 5,000 character deletion that happened. And with these 5,000 characters getting deleted, the impact, and this happened in both copies actually, and the impact was absolutely strange. It's called heterotaxy, where typically your liver is on your left and your stomach is on your right, and that got flipped strangely. So very strange things can happen with, some innocuous characters getting deleted or flipped or changed, etc. So the first application, and this is not for preventive health, but I want to give you a sense for 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 this, because this is something now that we do day in, day out, right? Once somebody has a disease like this, they come in saying we need to get our genome sequenced. We sequence the genome, we look at the rare variants, and then we try to pin down which of these rare variants is causative of the problem. Typically, the challenge that we have is that you have tens, if not hundreds, of these rare variants in every individual, most of which may not have an impact. But that one or two that could have an impact is an art and a science and an industry to get to the bottom of at industrial scale. And a lot of computing, a lot of literature analysis, a lot of medical expertise goes into that eventually to getting these few characters that might be problematic. And that's a industrial scale thing today, right? Lots of people are born like this. It's rare, but one in a few hundred, one in a couple of hundred people might have something like this. And then that's a large number in a country like India with 25 million births. And so many people have this. They come in to get sequenced so that we can identify their DNA, their variant, specific causative variant. And how do you use this now, right? The way it is used is that somebody wants to have their next child. The first child has this problem. How would they have their next child? I mean, imagine the unease that you would have knowing that this could happen all over again. But now we have this tool. Right now you can sequence, you can figure out what is the character that's problematic, and you could test your next pregnancy in utero, so inside, while the baby is inside the womb. You can test it, and there are ways to do that, to see whether they carry the same character or not. And if so, there's an option to abort. So that's a powerful tool that's come about in the last 7 years, and that's scaling up and becoming really commonplace, right? I mean, given that one in a couple of hundred people might have to do this in a disease setting. Of course, what we want to look at here is more in a preventive setting, and that's where I want to come to. But before that, I want to mention that in a disease setting today, we are able to solve about 50% of the cases that come our way. We do this on an industrial scale. And 50% of the times we are able to point out, here is the variant convincingly, and provide a tool to these families to plan their next pregnancies. In maybe another 10, 15, 20% of the time, we are able to say, look, we found something, but we are not entirely sure. That's a very uncomfortable situation to be in. You have a serious decision to make, and you're saying this might be the cause, but I'm not entirely sure. And there are doctors and patients handle it in various ways, but what I want to convey to you is that we are still evolving. This is not a perfect science yet, but the fact that we can solve 50% of the cases and give 50% of the family the tool is tremendous progress over the last 7 years. But let's come to the preventive setting now. In the preventive setting, what can we do? And this is what I meant earlier by saying that while rare variants are rare, from the perspective of an individual, they're not so rare. So 1/3, in fact, it's 35 to 40% of the individuals. Again, sorry for the bad English here, slides made in a rush, but 1/3 of the individuals, 1/3 of all of us would be a carrier for a rare variant that can cause serious disease. Okay, so what does that mean? It means that, there's no impact on the person, the carrier themselves. So I might have be a carrier for a rare variant. I would see no effect at all. I'll be a silent carrier. But if my spouse is also a carrier for the same gene, then each of our children would have a quarter chance of being affected. And what do I mean by serious disease? What types of diseases are we talking about? This is a list of sort of most common. We just took the last 80 cases that we did and said, what is showing up? Towards the top, you'll see something called beta thalassemia. 1 in 25 people is a carrier for beta thalassemia. Now, if you do the maths, if 1 in 25, if people are marrying randomly, 1 in 25 into 1 in 25 is a chance that 2 carriers mate. So 1 in 25 into 1 in 25, 1 in 6, 25, let's say, and then a quarter chance that a child, so 1 in 625 into 4, so 1 in, let's say, 2,500 or so, would be the chance that you would have somebody with thalassemia, right? Unless you do something about it, unless you prevent it in some form. So, and beta thalassemia is a disease where the mutation appears in the hemoglobin gene, the gene that the hemoglobin, we all know, is the protein that carries oxygen to our tissues. And if something goes wrong with that, our body cannot get enough oxygen, which means, serious consequences across the body. And so people with beta thalassemia need to have frequent blood transfusions, get blood from somebody else so that that hemoglobin can carry oxygen. So that will allow them to carry oxygen, but then it's somebody else's blood, and it has its own set of things that lead to problems, and frequent blood transfusions and all, not easy to do, right? I mean, we're getting better at it, but it is a condition of morbidity that leads to significantly reduced life expectancies, and quality of life, not to say. And so, for example, you'll see cystic fibrosis here. That's a condition where your lungs get clogged and leads to lung disease. That there's glycogen storage diseases. This is where your body lacks a particular enzyme, and in the absence of that enzyme, progressively, very early, like even by the time you're a few years old or getting into your teens, 20s, definitely, your body will get into serious challenges, and often these kids can die in their teens, 20s for sure. And the only therapy available is, the enzyme replacement therapy. You have to replace that missing enzyme, and that is extremely expensive and not easily accessible. So these things are, serious concerns. These are not easy to manage. And the fact that 1 in 3 people is a carrier is a significant statistic. I mean, for us as a society to take note of. Any given individual, the chances are, even if I am a carrier, what are the chances my spouse will have a mutation in a variant in the same gene? Pretty low. But at a population scale, things happen, and we are seeing that through the disease spectrum of referrals that come to us saying, I had one child with this. How do I navigate around that? Right? And no, there's no way to know whether you're a carrier other than sequencing your DNA. When we noticed that as many as 30 to 35 to 40% of people were carriers for one of these diseases, we were a little worried that, is it a statistical artifact? What's going on? And so we've been monitoring this. We take every set of 80 or 100 samples and say, does that statistic settle down? So any set of 50, 100 samples you take, eventually it settles down to between the 35 to 40% mark, people who are carriers. So it's pretty robust, I think. And that's a large, presence. So this is one key application we see as something that can reduce the total morbidity that's out there in terms of number of kids being born with these diseases. And it is in our eminent interest as a society to reduce that number. The problem, though, is that the action to reduce that number has to be taken not by the people who are suffering from the disease or their families, but by pretty much all of us, because we never know who's a carrier. And therefore, it's broad scale screening that needs to be done to reduce these numbers. So that's one clear application. I want to move to the next preventive application, which is determining cancer risk completely. And that is let me ask a little question. I know we can't gather answers, but maybe you can type in these answers. How many of you recognize, or how many of you do not recognize the person in the center? I know that everybody would probably recognize the one on the left and the one on the right, given that they are they are popular celebrities today. I don't have a way of seeing the answer. Perhaps, Shweta, you can point out if a number of people say that they don't recognize the person in the center. But I can give it a few seconds and I'll tell you the answer in a little bit. But these are all famous people who have, who either they or their parents carry a variant in the BRCA1 or 2 gene, which I referred to earlier, which renders them at a very high risk for cancer. Angelina Jolie, of course, has blogged about it extensively. You can read her blogs, and she had her mother, aunt, everybody died of breast or ovarian cancer in their 40s and 50s. And so it made sense for her to ask the question, what can I do to ensure that doesn't happen to me? Beyonce's now's father, you don't think of males having breast cancer, right? But her father has breast cancer because he has a mutation, a variant in this gene. And males can also get breast cancer if they have a variant in that gene. I'll come back to who the person in the middle was. Maybe at the end we'll come back. But the question then arises, right? If these sorts of variants can lead to high risk for cancer, should we be testing for those? And there's been much debate about population-based screening for variants in these genes. And this is the discoverer of the BRCA gene. It's called the BRCA gene. Mary Claire King is the discoverer of this gene. And she's been a vocal advocate saying, it's time to really offer screening of these genes to every woman about the age of 30 in the course of routine medical care, because this can potentially reduce the total burden, because that's often, it's not necessary that there be a family history in many of these cases, particularly if it goes through the paternal route. Your father has a mutation, a variant, and it comes to you as a daughter, while your father would be had a higher risk for male breast cancer, but male breast cancer is very rare, right? So it's very rare generally, even with your risk goes up substantially if you have a variant in the BRCA gene, but it's still relatively rare. So you may never know that it's in the family. And again, family histories may or may not be maintained. What happened in our grandmother's times, may not remember. She may have passed away of something that was generically described as something, but could have been breast or ovarian cancer, etc. So it's not easy to determine who has this variant. And that's therefore the call for broad screening, right? And now the question is, one has to do this rigorously. And can one save lives by screening? And there's health economic studies that have come out that show, and what does a health economic study do, right? They say, if I were to test everybody, then it's going to cost me money. It's going to cost me, meaning depends on who pays, if it's the government paying in like a European system, or if it's a insurance company paying in the US, or it's an individual paying out of their pocket in India. Somebody has to pay for the cost of the test. And is it worth that cost? What you gain from that knowledge, a large number of people will get tested. A fraction of them will be positive. Will that fraction be able to do enough so as to extend their lifespan, save their lives, or reduce the onus of disease? And since the economics in different countries is different, the cost of treatment is different, the success rates of treatment may be different. So these studies are done on a sort of a country by country basis. And studies have been done specifically focused in India, this particular study, for instance, and it shows that if you approximately about 800,000 breast or ovarian cancer cases in India, if you take the entire pool of women in India and everybody above 30 were to get tested, then 800,000 breast or ovarian cancer cases in India can be prevented. These people will go on to 800,000 people who would go on to develop breast or ovarian cancer would not get cancer. And some number of lives, maybe a few 100,000 lives, can be saved from succumbing to these diseases, right? So so the economics is starting to point in the right direction, and the outcome studies are starting to point in the right direction, and the cost of sequencing is really reducing, as I said, right? And so that leads to this interesting situation where one can use this now as a tool to control one's risk of cancer. Now I must be careful here and ask the question, right? What fraction of cancers arise on account of these monogenic conditions? And that's where I want to give you a few different statistics at this point of time. One statistic I want to give you from, if, let's say, in our last 180 cases that we looked at, asymptomatic individuals tested, we found 6 instances where they have where they have a variant which increases the risk of cancer substantially. So let's say, roughly speaking, 3% of individuals will have a variant that increases their risk of cancer. So if you test 100 people, maybe 3 will be positive, of which the 2 key genes, I've been referring to constantly, the BRCA genes, is about 0.75% of people will be positive. And then there are other genes that are mentioned here that also provide increased risk of cancer, albeit to a slightly lesser extent than the BRCA genes, but yet substantially. Okay. So this is one statistic to keep in mind. 3% of people or thereabouts, and you can take this number with a certain fuzziness. Maybe it'll be 2.5%, maybe it'll be 2%, maybe it'll be 4%, but not a whole lot lower than that. Let's take an average of 3% of the people will be found with a variant that causes an increased cancer risk. Now, what can these people, if I have a variant that leads to an increased cancer risk, what can I do? Right? It depends on the nature of the cancer, and there are guidelines typically for screening oneself. For example, for breast cancer, the American Cancer Society guidelines say that everybody between 40 and 44 should have an option to start screening with a mammogram every year. So women between the age of 40 and 44. But once you get to 45, until 45, you sort of take your pick. But until 45, you better do that every year. Beyond 55, you can reduce your risk a little bit, because reduce your frequency of mammograms every year, because every testing methodology has mammograms have radiation that can cause, over a period of time, increase your risk. So when you want to balance out those risks, your risk with age is going up, and you don't want to you can reduce anyway. So you're going to get cancer, as your age goes up. So switch to mammograms every other year, and should continue as long as you're in good health, let's say, right? This is what your guidelines typically say if you're the average person. Now, if you have a mutation in you have a variant in one of these genes, you're one of the 3%, let's say, then the guidelines change. So they say absolutely consider annual mammogram starting at age 40. No, don't wait till 45. And consider a breast MRI with or without contrast. So not just mammograms, consider an MRI starting at age 40, because that has advantages. At between 45 and 50, consider risk-reducing surgery. If you remove your ovaries, then your chances of ovarian cancer go down, right? So these are all in the guidelines today, right? And this is for this particular gene. If you remember on the previous slides, I had a few examples in our cases. So this particular gene, RAD51G, when I look at the guidelines for that, that's what they say. So there is an evolved set of guidelines gene by gene saying if you find a variant in this gene, then this is what you need to do, right? So there's clear actionability with some amount of evidence driven from studies that have been done. So the first part of the statistics I wanted to convey is that, let's say 3%, you can take that, as I said, with a pinch of salt, maybe 2%, maybe 4%, but somewhere there of people in the population, in the general population, carry a variant that will put them at an increased cancer risk. And for them, typically there are guidelines that can help, that have been proven statistically to help reduce that risk. But now let's ask, right, what percent of cancer is caused by these monogenic rare variants? Well, 3% of people have such variants, but what fraction of cancer is caused by such variants? So for that, you've got to look at cancer. Cancer is like a tumor tissue, right? And a tumor tissue has its own genome, interestingly. And this picture depicts a tumor tissue, and it has think of it as having its own genome. Over a period of your lifetime, even though every cell was born with the same genome sequence, over your lifetime, different cells have developed different variants, and they have become different. And particularly variants which enable a cell to divide faster are more likely to create more cells with different sets of variants, right? And that's the Darwinian way in which things evolve. So a cancer will have its own set of variants. So what we do in routine practice, and we do this because cancer patients come to us and they need to get the cancer genome sequenced so that they can understand what caused the cancer and whether that leads to any therapeutic implications. So certain types of mutations in the cancer can be treated with certain drugs. And so that's a whole science and medical practice in itself, which I will not get into because I'm looking at the preventive element of things. But what I'm pointing out here is, because cancer patients come to us and we do a lot of this cancer sequencing, we get to understand, and note also that a tumor is not homogeneous, right? There could be many parts of the tumor with their own genomes, and that's just something to keep in mind. We don't dwell on that much more. But what this allows us to do is to look at, all these people have gotten their tumor sequence from us. In what fraction of the cases did we see a variant that they were born with? There are ways to figure out what you're born with and what you've developed over your lifetime. So in what fraction of these cases do they have these variants that they were born with that would have rendered them at very high risk for cancer, right? You can ignore the caption, but broadly speaking, the takeaway is that ovarian cancer, almost 20%, the dark blue is what you need to look at, 20% of the tumors that we have tested and sequenced genomes of have a variant that they've been born with that has put them at very high risk. For prostate cancer, which of course is a male-only phenomenon, almost 14% of the tumors that come our way those people have been born with something that has put them at high risk. Pancreatic cancer, about 2% odd. You can say that's a small number given that, but breast cancer, about 10%. Colorectal cancer, about 3%. Right? So if you take, colorectal, breast, prostate, ovarian, a minority, but significant fraction of these cancers today are being caused because you're being born. You're born with these mutations, with these variants. And if you test for these variants, as I said, there are guidelines that will help minimize the risk once you know that variant is there. And so that's the really the point. Now, you could ask, how about the rest of the, even with ovarian, there's only 20% of people who, 20% of the cancer patients seem to be born with something that rendered them at high risk. How about the rest? Now, that is unfortunately the reality of life today, that age is the biggest risk for cancer, because as you age, your cells develop variants over a period of time. And as that happens, you get your genome changes. And even though you were not born with a problematic variant, you will develop one with age eventually. And just in these two genes, we found that while 20% of people were born with those mutations, another about 5% had developed problematic mutations in just these two genes. Sorry for the noise at the back, it will die down in a minute. But yet that still leaves the remaining, right? Why did the rest get cancer? Well, that's the open question. There are many, many genes. There are many other phenomena. Today, where genomics is, we are now in a good, solid scientific medical position to chip away at, between 2, 3, 5 to 10, 20% of the disease burden, but not much beyond that. But at least that we are now scientifically robust on that front. So let's look at how reliable is all of this. Okay. The process is, I want to tell you this one thing, that this is a very complex process, meaning that sequencing today requires maybe a large number of steps in the lab, a large number of steps on the data analysis, the business rules needed to figure out which variant is problematic, which is not. Every person has a large number of variants, most of which are not problematic. You need to understand what's there in the entire medical literature, all of medical knowledge, clinical knowledge, biomedical knowledge, computing, software, lab, a whole lot of things have to come together. But the beauty of the process is, it's, when done properly, it's extremely robust. You can move it from one lab to another as long as you follow the steps with rigor, and as long as you have really good people who understand the process. The process is extremely robust. And most variants found by sequencing, and you cannot say the same of the microarray technology, but most variants found by sequencing are beyond doubt, right? But there are challenges. There is so much unknown today. So as I said, when a person with disease comes to us, we're able to solve those cases 50% of the times. But in 20% of the time, we'll find these variants, but we don't know what those are. I mean, whether they are problematic or not. We have a suspicion that they are, but we're not able to convincingly prove the case. So there's so much unknown. There are large parts of the genome, think of the genome as a large set of real estate, where some parts we built buildings and we mapped out cities, and we know those places very well, but there are large stretches of wilderness that we haven't explored. And so there is, any individual's disease could be coming from those parts, and we may still not have explored those parts, and so we won't be able to get an answer there. There are these more complicated variants where large chunks of the genomes get deleted or duplicated or get moved around in complex ways. Those are not always easy to catch. And we do a lot of hard work trying to figure out, when we suspect a variant like this, it needs a lot of thereafter diligence, and something that cannot be put into an assembly line, but where smart, good people have to stare at it from various angles and convince themselves whether this is really a true one or not, and whether we've caught it or not. So, a fraction of causative variants can be missed yet, but the process is so robust, as I said, to catch a good set of things that are now today useful, medically actionable, and can save a fair amount of a reasonable fraction of morbidity and mortality all around us. I'm going to end in maybe 2 or 3 minutes, where I want to give you a glimpse of what is coming up in the future. So everything that I talked about today is out here, available today, here and now, right? The world has gotten to a point where we can determine who's a carrier. Carrier screening broadly can help reduce, hopefully, the overall disease burden by a significant amount. We can find a fraction of a fraction of cancer cases between, let's say, whatever, 3%, 20%, which are caused due to inherited mutations that one is born with, can be caught through broad screening, and action can be taken to reduce that risk. And there is enough health economic justification for that. But now all of that is to do with what one is born with. I want to now look at what I mentioned earlier, right? That age is the greatest risk factor. And with age, different parts of the body are going to develop their own genomes. They're going to develop variations which are going to, their genomes are going to just evolve with age. And how do we, while we can measure, you can do a one-time lifetime test, once in a lifetime test to sequence what you were born with, what is evolving in your body is not a one in a lifetime thing. And how will we ever figure out how the body is evolving, and do we have a way of doing that? So this is on the cusp of sort of coming out into practice, and you'll hear more and more about it in the months and years to come. But the fact is that from a drop of blood here, not a drop, you need a significant amount, let's say 10 ml or 20 ml of blood, which is what you would, which is a little more than what you might, or maybe comparable to what you would give for your annual biochemistry test, 10 to 20 ml of blood. What happens in the blood is there are these free floating DNA fragments sitting in the blood, and they come from all over the body. So now that gives you a clue, right? If DNA in different genomes in different parts of the body are evolving in their own way, all of them, in all those regions, the cells are dying, there's some natural turnover, they will let their DNA loose into the blood. And now we can trap it from the blood and sequence all of this DNA. And that will get us a window as to what is happening all over. And of course, the challenge is that this is such a diluted signal. If there is a cancer lurking inside or something else lurking inside, that will be such a small fraction of cells that the DNA that leaks through from here will be such a small fraction that it's a needle in a haystack problem, right? You have to be really, really very sensitive to be able to catch this. But we've gotten that today. There is DNA sequencing. There's also something called epigenetics, which we'll perhaps discuss in the discussion session, so I'll not go through it. But I want to end with this one slide. We've been working on this, and we now have this study completed where we can show that we can, from your blood, from just 10 to 20 ml of blood, by looking at the DNA that's floating around, we can detect in a fair number of cases that there is a colorectal cancer lurking inside, right? In 65% of stage 1 cases, early stage cases, and greater than 85% of stage 2, stage 3, stage 4, look at this dot, the median value, that's 85%, with about a 10% false positive rate. So specificity of 91%, we can tell you that there is a colorectal cancer lurking inside. So these are interesting emerging technologies that use epigenomics that will come out, that have been coming out in the very recent past, and will be talked about more and more in the future, that can help us get a sense for our evolving genome. But for today, our inherited genome is completely, the tools are there today for us to use to the best benefit of society. I'll thank you with that, and I'll invite Shweta again to perhaps lead the discussion.
Thanks, Ramesh. That was really wide-ranging look at genomics in health. Just for the record, there were 4 people who didn't know who the woman in the middle was. Did you want to talk about that?
Okay. So, and I'm assuming all those 4 are relatively young, and so that's Chris Evert. She was the Wimbledon champion many times in the late 70s, when I was growing up, in the early 80s, perhaps. She too has come public with the fact that she was actually maybe in her 50s. I'm not sure of what age she was when she went in for just a preventive checkup, and as part of that, they discovered that there was a cancer in the ovaries, I think, and got tested at that point and found that she was actually positive for a variant in the BRCA gene.
So there's a few questions about, the evolution of technologies. So you started with microarrays, moved on to sequencing, and sort of stopped with how we're doing relatively well at identifying germline or inherited variants. But there's a few questions about long read sequencing. So sort of asking, how is this still evolving? What are the newer technologies that are arising? And at what point do we expect long read sequencing to become the standard compared to what we have right now?
So as I said, to set the context for the question, one of the things that I'd mentioned is that the only way we can sequence genomes today is by chopping them up into tiny pieces, which are, let's say, 150 or so in length. And that leads to problems because to assemble that jigsaw back, I need to figure out where each piece came from in the reference sequence. And that's not always easy because sometimes there are sections of the reference sequence that repeat. So in our genome, the sequence is such that there are chunks that repeat. So now for any given shred of paper, I don't know whether it came from here in the reference genome or it came from there. And that leads to lots of complexity. And even in a real disease context where we find a variant in certain genes, let's say there's this gene called PMS2, if you find a variant in that, it's a nightmare because that whole gene has an almost identical copy of it elsewhere, and we have to be sure whether it's the variant is in the gene or in that other almost identical copy, which is not really doesn't really play a role in disease. So these things become a nightmare, and then having a larger, your shreds of paper being bigger instead of 150 maybe being 1,000 or 10,000 or 100,000 would be tremendous, and that's what we call long read sequencing. And the challenge with that is that it's very expensive today to do long reads, and it's also more erroneous. Like every process, the sequencing process has error, and I didn't talk much about it, but that error is controllable, and we do things to control that error. To control that error, you need to essentially incur cost. You need to read the same character many, many times. And with long reads, errors are more, you need to read more, so you get help in a way, you get more challenging in a way. And so if we could get to long read cheaper, then it would be tremendous. The standard 150 length things are getting cheaper, and as I said, becoming very, very accessible. So I see the next few years, next at least 2 or 3 years of medical practice adopting, largely driven off these short 150 length things. And then hopefully over that period of time, maybe costs will come down further for long reads. And eventually we know that everybody's guessing that technology will reach a point where we will do 10,000 length shreds of paper as opposed to 150.
You also spoke about sort of both the potential of genomics in health and also areas where it's limited, its applications are more limited, and perhaps plenty of an area in between where it's not quite clear. And in light of both of these things, so firstly, the potential, there's a question about, do you see a point of time where this preventative screening is done, at right when a baby is born? How widespread is it going to be? I know in the UK, they are already talking about newborn screening for preventative care at a population scale. Do you see that as standard practice in the future at any point?
Yes, so broad scale screening, right? So I talked about 2 applications of broad scale screening, maybe a third emerging one at the very end, but 2 I talked about. One is carrier screening, where you might be a carrier and which might render your baby at risk. And the second is inherited cancer risk. Those are both, I think, in different countries are in different stages of adoption, and economics are different, but medically, technically speaking, both of those are, as scientific tools, are eminently available, and it's a matter of just maturity, time, adoption, that more and more people will adopt that, and screening will become commonplace. There are other forms of screening that you point out which I have not mentioned, so I'll go through a few of those. So newborn screening is one thing that we talk about quite a bit. What happens is a small fraction of babies end up in the NICU, the neonatal ICU, with a severe condition. And It's a very small fraction. But the point is, that condition may develop. It may become apparent a little bit later in the first several days, or it may take a little bit of time. And you have a very short window to act. And if you act in that window, then you can arrest things. If you miss that window, then there's a problem. So with that in mind, I mean, since you don't know who will get into that situation or not, people have been exploring in the West this notion of newborn screening. And that needs to be done very rapidly, right? The window is very, very short. So you need to do it in like 48 hours or some very small amount of time. Sequence everybody. Most people, you'll find nothing, but there'll be a small fraction of people, as usual, where small fraction of babies, where you'll find something. And if they start showing symptoms, then you'll, or even in advance of that, you'll be able to address their, with supplementing it with the right enzymes or whatever is needed. And so that has led to this notion of newborn screening. The studies on this are, hot off the press, so to say, like results have been coming out over the last year and a half, maybe. And there is a case to be made for that. The economic case on that is yet to be studied, except in the West, it's been studied. So wherever the average cost of a baby is several thousands of dollars. So people have done these studies in the West. They've shown that newborn screening is very effective, because if you get into trouble, then that cost is like hundreds of thousands or millions of dollars. And if you don't, then it's nothing. But now the trade-off sort of works with what fraction of babies get, need eventually benefit from it. In India, it's a little unclear that those health economic studies have not been done, where the benefits of screening versus the lives you would save, etc. So it then becomes an individual call. And that's why I sort of focused on areas where there is that health economic impact there. Now, to talk about a few other areas, right? Let's talk about all these other complex diseases that we as a society struggle with, right? Obesity, hypertension, diabetes, dyslipidemia, heart, coronary artery disease. It's a large set of things, and all of these have a lifestyle component, a genetic component. Of the genetic component, there is definitely a monogenic component to it. It'll be a few percent, or smaller in some cases. Diabetes may be even smaller. Age-related diabetes, there's a younger age version of diabetes which is highly monogenic, but so there are varying fractions, and there is value to in some of these to determine if you have a monogenic variant that puts you at increased risk. It is less clear what to do in that situation in many of these. It is in some cases, I mean, for example, there are heart malfunctions that electrical malfunctions or structural things in the heart that are silent that you could benefit from knowing. So there are, I didn't talk about all of that because the science on that is a little less evolved, and I've taken the most clinically well-proven, real-ready, sort of well-studied, and brought those out. But there are definitely areas there where there is benefit, but it needs a longer conversation.
There are actually people who are interested in knowing where they can get their genetic testing done in India. And for example, like, would Strand have this preventive cancer screening that you spoke about? So could you say a little bit about that?
Yeah. So over the last, I would say 5 to 7 years, genomic testing has evolved substantially in India. I mean, it's heartening to note that I would say we started our genomics lab in 2013, and we got a College of American Pathologists accreditation in 2014 or 15. And at that point of time, there was nothing at all available, right? Very little. A few research organizations would do a few tests on in very specialized situations. From there, we've come a long way. I mean, the country has come a very long way. I mean, at Strand, we offer broad we do large numbers of tests. Most of it is people with disease coming and requesting sequencing, but increasingly, for all the reasons that I've mentioned in my slides, and there is a case to be made for the asymptomatic person to preventively get tested. And you can just reach out to our Strand website, for instance. Go to this for the asymptomatic, the regular healthy person, we call it the Genomic Health Insights test. If you go to our website and go to the Genomic Health Insights, navigate to that, you'll be able to learn more about how you can get tested.
Maybe I'll ask one more question before passing it on to Sushila. But there's a few questions about the sort of social, policy, ethical side of things. Until we have a clear idea about where genetics can and cannot help. What do you think about the misuse and misinterpretation of genome sequencing? So, for example, the use of genetic data by insurance companies, or let's say for targeted advertisements. Are there conversations about that happening in India, and are these things that people who do get genetic tests should we be aware of those?
Yes, so that's a very important aspect, and there are a few different elements there. I mean, the first element is it's a complex field, and who can interpret these results? Can you do consumer test and trust what is being said? Can you interpret and take action based on it? So what is amply clear to all of us in the field as scientists is that it is a complex area, and that while we do, while I believe there's a lot of value in, because in India, the healthcare system is never going to be able to pay attention to your, as an individual, to your health. You have to take care of your health, right? So there is a lot of value for you to take care of your health by reading and taking things into your hands. But any action has to be absolutely, it's a complex field. Any action has to be taken by a medical professional who has to look carefully at what was found, who did the test, what did the report say, was all the regular due diligence applied, and justification, robustness, etc., and then come to a conclusion, right? So while medical tests, while consumers should, I do believe, because I, as a person, nobody else will be thinking about my health. I do need to, take whatever I need to do, get whatever information I need to, but the interpretation of that information and the actions around that absolutely should not be done by an individual themselves. So that's one element of it, because there can be a lot of, there will be lots of people offering to, sequence, offering to provide reports, offering to say that you have this disease, that disease, or you do this test and your fitness can go up, or you should eat this sort of food and all of that. There is likely to be a lot of misinformation and all of that, and it's got to be done very carefully. So that's one element of it that we need to be careful about. The second element of it that we need to be careful about is, the stigma associated with this. I find that I am positive for something, I might be at a higher risk for something. As a society, we don't understand these things very well. We don't understand that the best way to live life is to understand and act on the risks rather than ignoring them, and or by judging people who have risks. And that's so privacy of information has to be absolutely paramount, which means that your genetic data is yours and is not seen by anybody else. If it is seen by anybody else, it has to be in absolutely watertight anonymity, with research consent, with everything, that ensures that there is no chance of it being linked back to you. So that is something that has to be practiced. And there are various ways, both, the legal data protection end of things is evolving. The clinical research guidelines that mandate all of this, the companies themselves putting in place enough safeguards on the ensuring that access to that is very, whatever multi-factor authentication, security safeguards, etc., etc. Then the third aspect of it is, suppose you are at a higher risk, right? Can you be discriminated against by insurance companies, and can they say that we will deny you insurance? This is an evolving area. In the US, they have tackled it by making a blanket law which says no company can deny you health insurance on account of your genetic results. That's a blanket law that's there. So that protects and that opens up the field, saying at least, I'm not going to be denied health insurance. In India, that doesn't exist. It's evolving in some sense. There's somebody is challenged, somebody is gone to court. There is something, I think one of the courts ruled in favor, saying that the insurance company cannot discriminate, and then there was an appeal in the High Court that's pending, and so there is something going on there that's still pending. But in short, I think what insurance companies need is, they need you to declare your pre-existing conditions. It's not clear, and it's probably not true that one would label this as a pre-existing condition, because there is no condition per se. I mean, these are things that up your risk, and they've not led to any condition per se. So there isn't, perhaps insurance companies should not know about this for sure, and there's perhaps no need for one to reveal one's condition, this to an insurance company. And hopefully we will have a much more formal structure, which we all need to lobby towards, which will open this up much more formally.
There are many questions that we couldn't get to, but I think we will have to pass it back to Sushila. But thank you, Ramesh, for that very sort of comprehensive discussion. And I think one of the most exciting things about this area is how multidimensional it is. It's the kind of challenge that has to be addressed by multiple stakeholders. And I think that's why it's so important that we have these conversations with people whose lives are likely to be affected by genetics in the very near future. Sushila, would you like to take over?
Thank you. Thank you very much. We've had yet another very exciting and interesting webinar today. Thank you very much, Ramesh and Shweta, for today's webinar. Just want to run over a few key takeaways. We started today with Ramesh explaining to us the 6.4 billion characters in each of us, in the 23 pairs of chromosomes that of varying sizes that we have. And he talked a little bit about what makes each of us different genetically, both in terms of physical characteristics and in terms of disease predisposition, etc. We looked at a few specific genes, such as BRCA1, to explain what happens when a gene gets modified. Sometimes there are very serious consequences as well. And then we looked at the history of genetics and what has brought us to where we are today. In approximately the year 2000, when the first human genome was published, I still remember the announcement and the excitement that all of us felt around something like that happening. And so then we had a reference genome sequence that existed. Until the year 2015, the technique that was used to read genes was the microarray technique, but this allowed probing at specific parts of the gene. Over time, the cost of that came down from several $1,000s to about $100. But still, there were a lot of gaps in terms of what that reading actually told us. Obviously, the complexity of this whole area is extremely high. And we saw an example of that when Ramesh talked about autism and how the rare variants result in about 6 to 10% of risk. But the common variants are what account for 50% of the risk itself. So it is a very complex area. Like he said, there are a lot of bits and pieces of research from across the medical community that needs to be interpreted, brought together to make sense out of it all. But since the year 2015, the sequencing technique that has evolved has also become very robust, and that is what has made it all so exciting and made us able to think about various applications, especially in terms of prevention of diseases. And this has been enabled by, Shweta mentioned, very multidimensional, so very multidisciplinary, so things like molecular biology, computing algorithms, optics, etc. Many different pieces have played into helping this technique work. We can also do a lot by sequencing the relevant parts of the gene rather than reading the entire gene. And I thought that was an important thing to take note of. And then Ramesh spent most of this talk was about preventing disease. And so he spent a lot of time talking to us about how genetic sequencing can actually help. So while there are clear guidelines for screening an asymptomatic person or a healthy person, if there is a particular genetic mutation that is noted, then what are the guidelines that apply? And in many cases, in many areas, that too is now in existence. He talked about tumor sequencing and what has been learned from that. We are born with a certain risk of some certain diseases, but over time, as our bodies age, we may develop additional mutations. And this again changes the probability of various diseases that we might contract. So, it's not like we are born with a certain stack and we can just conclude that our lives are made and we can go ahead and eat and drink what we want and live a stressful life. So irrespective of what the genetic sequence says, we still have to focus on our lifestyle and all. There's no getting away from any of that. We learned that this is a very complex area and it needs a lot of care in interpreting the data. We also learned that there are many, many unknowns still that we need to work through. And then we looked at how, particularly at detecting cancer early, and what are the things that we can look forward to going forward. And he talked about liquid biopsy from a few ml of blood and how free-floating DNA, mRNA, etc., can be sequenced, which will give an indication of an existing problem somewhere in the body. He talked about epigenetics as an emerging area. And then he talked about the possibility of colorectal cancer being detected non-invasively. So those are some of the things that we can look forward to. I think there is a lot more happening, and things are happening on a daily basis, as I understand. So just to wrap it up, we had a short discussion around the ethics of all of this. So thank you very much. This was really very informative, an exciting topic, and I'm sure many, many of us have gained a lot from this session. For our audience, thank you ever so much for being part of this, for your questions, which have made all the difference in terms of the quality and the quality of the discussion that has happened. Of course, many, many thanks to Shweta for having been part of this, and Ramesh for your wonderful, very clear presentation today. This presentation, like all of our other webinars, will go up onto our YouTube channel. Please do look at it, and also please feel free to share it with anyone that might benefit from this webinar. So once again, thank you very much, and look forward to seeing you all at our next webinar.
Thank you so much. Yeah.
Bye.
Bye.